process. It defies classification in that it's very hard for us to look at a developing human and say, yeah, at this point, it becomes a human. Yeah, at this point, it becomes this. Yeah, at this point, it becomes that. We say embryonic development occurs for the first 8 weeks. That's largely because all the organs are made within the first 8 weeks. The designation is a little bit arbitrary, but it's useful for the science of embryology. Because of this continuity of development, essentially, when does this entity become a human being? It becomes a human being when it begins this journey of development. And that journey begins at the end of fertilization. So while this type of technology is a remarkable advance, it does destroy human embryos that would give you a baby. This is what an embryonic stem cell culture looks like under an axiophot microscope. You see these other little spindle-shaped cells crawling around. These cells are grown on feeder cells. These feeder cells are derived from animal cells, and they are usually treated so that they can't divide anymore, the feeder cells, that is. The feeder cells make a variety of growth factors, and those growth factors keep these embryonic stem cells from differentiating. It's possible now to grow embryonic stem cells and derive them without animal feeders. And you can either use human feeders, or you can use conditions that are completely feederless, because the growth factors that these cells make have been isolated. You can simply use those soluble growth factors, and now you can derive these without using animal feeders. You don't want to use animal feeders because the animal feeders will put proteins and carbohydrates on the surfaces of your cells that will cause them to be rejected by our own immune systems. Also, they may transmit animal viruses. That's another reason why you would want to use animal feeders. So the first person to derive embryonic stem cells was James Thompson at the University of Wisconsin at Madison. And as you can see, you can differentiate these cells into derivatives from all 3 embryonic germ layers, ectoderm, mesoderm, and endoderm. So you can make digestive system type cells, you can make nerve cells, bone type cells, cartilage cells, muscle cells, kidney cells. In all those panels there, you can see we've hit all 3 of the basic embryonic germ layers. And the usefulness of these cells really cannot be overstated in many ways. But when you make them, you do destroy a human embryo. Now, most people, when they think of embryonic stem cells, they think, well, we can use these to cure liver disease or muscle atrophy or Alzheimer's or something like that. That's just one of the many usages of these types of cells. Believe it or not, one of the biggest uses of these cells is actually in drug screening. Because you can take these embryonic stem cells and differentiate them into kidney, liver, gut, whatever, you can determine how that kidney, liver, gut, whatever will react to particular drugs. What sort of model systems did we have before embryonic stem cells? Rodents. Are rodents good model systems for drugs? Depends on the drug, depends on the tissue. When it comes to heart medicines, rodents are terrible models. The rodent heart beats very, very fast compared to the human heart, and it does not react in the same way to drugs, the heart drugs, the way the human heart works. So by using actual cultured human heart cells or human liver cells, you can actually use these cells for drug screening and get a better idea of what kind of adverse effects you might have and what kind of efficacies those drugs might have. You can also use this for certain types of gene therapies. As you grow these cells in culture, you can put genes into them. So as laboratory models, these cells can be very, very useful. But the downside is that they involve the destruction of a human embryo. Well, if you want to make stem cells, embryonic stem cells that is, that have the same sort of cell surface proteins as your own body so that they won't be rejected when they're injected into your body, you can use cloning actually to make cloned embryonic stem cells. And in this procedure here, you take an egg from a volunteer who has undergone a rather rigorous procedure to donate her eggs. You take the nucleus out of that egg, and that's harder than it sounds. You have to take the egg and you have to squeeze it just right, A little too much pressure, it goes kerplunk. Not enough pressure, it's not going to work. You then take a small little micropipette, and it sounds like this takes a steady hand, that's because it does. You then suck the nucleus out of it, and then you take a cell from your patient, and you fuse it by just giving a little jolt of electricity. And now this cell will start to divide, and it will, if all things considered equal, form an early blastocyst, and you can then take that blastocyst and make embryonic stem cells out of it. Those embryonic stem cells then can theoretically be used to treat your patient who has a bad liver, a bad heart, a bad, whatever. The problem with this type of technology is multifold. First of all, this process is immensely inefficient. Scientists have been able to get it to work relatively well in the mouse. What relatively well means around about 5% on a really good day and 2% on a typical day of these embryos will actually get to the blastocyst stage. Then out of those embryos that get to the blastocyst stage, only a small percentage of them will actually make embryonic stem cells. So if you want to use this procedure, you need lots and lots and lots of eggs. And how do you get those eggs? Well, you get young, healthy women to undergo these procedures where they take these drugs that hyperactivate their ovaries, and then those eggs are harvested. Oh, gee, that sounds dangerous. Yeah, it kind of is. A line that you'd make would be in the millions of dollars. And that's provided that the line is actually useful, because not all lines can differentiate equally well into the tissue that you want. Also, you have made an embryo. Oh, well, it's a cloned embryo. Well, so, you have made an embryo. You have made a young human being. And you've made it just for the purpose of destroying it. And opponents of this research call this clone and kill. And it seems to me that's exactly what this is, clone and kill. Your congressperson may write you a letter, and if you object to this, say, oh, this isn't reproductive cloning, this is therapeutic cloning. Reproductive cloning means that you're going to take that blastocyst and you're going to implant it into the uterus of a recipient who's taken hormones so that her uterus is receptive now to the egg. And if all things work out properly, she'll give birth to a bouncing baby boy or girl, or in the case of Dolly, bouncing baby lamb, or leaping lamb, or, yeah, lambs don't really bounce, do they? This procedure is extremely inefficient, and all the evidence we have in humans says that whatever the inefficiencies here, again, Dolly, we had almost 300 tries to get Dolly. It's going to be even more inefficient in humans. And I have a chapter in the book actually about why I oppose reproductive cloning, but essentially the bottom line is this. Why are you making a clone? You're making a clone as a replacement. Your genes are not the sum total of who you are. There are events that occur in development that actually change you into a different type of person. You could take you and rewind the tape, and you would actually become a different person. Dolly, in fact, had siblings, and those siblings were different sizes than Dolly and had different personalities from Dolly. Dolly was rather shy and retiring. Some of Dolly's siblings were very outgoing. So you want a replacement, but guess what? You're not going to get a replacement. Furthermore, this child essentially is being raised to be a replacement. You're putting that pressure on them to be someone else. Is that what we want for our children? I thought our goal for our children was them to be what they could, the most that they could be. But now we're telling this kid you have to be someone else. Think about that. Think about what that would do to a kid. Think about if that was you. If that was the requirements on you to be someone else. So this is supposedly therapeutic cloning. Folks, the only difference between reproductive and therapeutic cloning is what you do with the blastocyst. Do you destroy it or do you implant it? That's the only difference. So if you get a letter from your congressperson saying, oh, well, the bill I sponsored outlaws cloning. See, it outlaws reproductive cloning. They have just tried to pull the wool over your eyes. Don't fall for it. So how is the embryo different from this great examples of humanity I see seated before me? It's different in 4 ways. Size. It's very small. And in books they'll say it's no bigger than the period at the end of this sentence. It's different in its location. It is inside another human being rather than outside another human being. It's different in its extent of development, and it's different with regards to its degree of dependence. And that's a nice acronym, SLED, for you to remember. Let's take size. Does Shaquille O'Neal have more of a right to life than Julie Peters, who was in the movie Willow, who went to my church actually when I lived in Great Britain? Does Shaquille O'Neal have a greater right to life than Julie Peters? How about let's make Julie Peters even shorter. Does she have less of a right to life than Shaquille O'Neal? No. And some of you are looking at me saying, well, that's really dumb. Yeah, I agree, it is. It's dumb. It's dumb to assign a right to life to somebody on the basis of their size. So size has no moral relevance whatsoever. So the embryo is as big as the period at the end of the sentence, so what? So what? It's small. I have 3 girls. My oldest and youngest were very small when they were born. They were just over 5 pounds. Stacy was the big beefer of the group at 7 pounds. Okay, does that mean Stacy had more of a right to life than Emily or Rachel? No. And yes, that's stupid. That's why using size as a determiner whether or not something is human or not is really nonsensical. How about location? Okay, Maddie over here is our president of SAU Students for Life. Let's take Maddie and put her in McDonald's. Is she less of a person? Now let's put her in her garage. Less of a person? Oh, let's put her at SAU. Now she's really a person, right? Oh, so location doesn't matter? No. Location doesn't matter. And 9 inches of a travel down the birth canal doesn't make a fetus a human being. It was a human being before that. So location is also a nonsensical standard for determining if someone has a right to live. Extent of development. Do 6-year-olds have more of a right to live than 2-year-olds? Okay, 1-year-olds. 6-year-olds definitely have more of a right. No? No, some of you look at me, that's dumb too. I agree, that's dumb. The embryo is not mature, but neither is a 2-year-old, neither is a toddler. Degree of dependence. And it looks like I have a spelling mistake there, so for the English professor in the front here, Dan, I will just apologize. Degree of dependence. Many of you are very young, you're very healthy. Many of you will get older. You will become dependent. You'll need canes to walk. You'll need some of these, glasses to see. You may get to the point where you need oxygen. Are you less of a person? No, you're not. No, you're not. To say that they are less, someone who is more dependent is less of a person is essentially the strong essentially lording it over the weak. None of these differences, and folks, these are really, when you think about it, the only differences between an embryo and a human being. None of these differences are morally significant. Why are we destroying embryos? Well, because of the great promises. It's not a Maserati, it's a human being. You have to come to grips with if the embryo is not a human being, then what is it? It is a human being. It is a young human being. It's not a different kind of thing as a human being. It is a human being. It's simply a very, very young human being. We already saw that extent of development is not a morally significant category. Well, and I have several of the objections to the humanity of the embryo in here if you want to read more about it. And many of these, again, come from questions my students ask me. The embryo does not have a working brain. This is an argument from Michael Garzinga at Dartmouth University. Well, look, you define human death as a time at which the brain stops working. And the embryo doesn't have a working brain. Well, so the human embryo is really not a human life. There are big problems with this one. First of all, let's think about brain death. At brain death, essentially, your human being is not undergoing self-directed human functioning. That loss of self-directed human functioning is irreversible. The embryo, however, is in fact a self-directing entity. It is developing as completely self-directed enterprise. It is in the process of growing. It is not dead. A brain-dead individual is dead. The embryo is very much alive. So already we can see that this analogy really starts to break down. Also, Is there anything you can think of that's not human that is capable of growing a human brain? Yeah, the human embryo, but that makes it human. The embryo doesn't have a brain, but it doesn't have a brain yet. So now you're going to, really this argument becomes another one of those extensive development arguments. And it's a poor argument. Well, what about twinning? The embryo is not a single entity because it can twin. Folks, twinning occurs in about 0.5% of all cases. So you're going to say that all embryos, even though 0.5% of them show this ability, you're going to say the rest of these embryos are not individuals. Do you see the problem with that argument? Essentially, you could say, okay, look, 1 in 150 kids are autistic, so all kids have the traits of autism. What? Doesn't make any sense. And yet the rates of autism are higher than the rates of twinning of the embryo. So it assumes essentially that all embryos have that capacity to twin. That is not the case. The minority of embryos have that capacity to twin. Secondly, why does losing a capacity all of a sudden make you a human being? Think about that. The embryo is usually condemned for not being a human being because it doesn't have certain capacities, but now it loses a capacity, and now we're saying that that makes it human when it loses it. Oh boy, that argument really starts to sound funny, doesn't it? Thirdly, it's very clear from studies on in vitro fertilization that twinning is a response really of the embryo to damage. The embryo is repairing itself. One of the ways it repairs itself in response to certain types of damage is to twin. And it's also very clear that twinning is something that is genetically determined. So the embryo might actually have a twin in the embryo in a latent state. Now, if you're destroying the embryo, you're not killing one individual, you're killing two. What about recombination, this business where embryos can sometimes fuse? That's an even rarer event than twinning, and it also is very clearly a response to damage. So you're condemning the embryo essentially for fixing its damage. But wait a minute, fixing its damage is something a self-directed individual does. Self-directed individual, just like a human being. So really none of these objections really add up to much, in my view at least. And I think the embryo really is a full member of the human race. It's simply a young member of the human race. Does it have the right to a driver's license? No. Does it have a right to vote? No. But it at least has the right not to be harmed. Well, can embryonic stem cells really save us? There have been 3 clinical trials. 2 of those trials were pretty miserable failures. One trial consisted of 2 people, and it involved people with an eye problem. And in one patient, the injected eye got better, and so did the non-injected eye. In the other patient, the non-injected eye did not get better, but the injected eye got better. So you're 1 for 2 on that clinical trial. Not a very good, not a very impressive clinical record, frankly. Tumor formation. Now, tumor formation, you can get around it if you properly differentiate the cells. Those differentiated cells will now not form tumors, but purifying the differentiated cells from the non-differentiated cells is tough. It's very tough. So there are some publications that show these very rigorous procedures for purification, and the cells still produce tumors. So tumor formation, even though people will really slough it off, it is a problem. It's a very big problem. Now, while these cells will grow very well in culture, getting pure cultures of redifferentiated cells is not easy. So that's another problem with these. And also, it's very clear that growing them in long-term culture is problematic because they are not stable. They acquire mutations, and many of those mutations are the same types of mutations you see in aggressive human cancers. Well, so what are the alternatives? Well, it turns out in our own bodies, we have a veritable cornucopia of stem cells in a variety of places. Our bone marrow, our fat, yes, indeed, your brain, skeletal muscles, peripheral blood, your gut, and recently even a stem cell population has been found in the kidney. Also your teeth. There are stem cells really all over the place in your body. Many of these can be isolated. They can be cultured even to some extent. and expanded in the laboratory, they can then be reintroduced into your body in order to treat you of various diseases and conditions, what have you. In fact, over 73 conditions have been treated with stem cells from your own body and also stem cells from umbilical cord. Not just umbilical cord, but umbilical cord blood. So the umbilical cord contains the umbilical cord blood. The blood contains a stem cell itself that can make blood cells. However, there are other types of stem cells that are from the cord tissue itself. In particular, a little critter called a mesenchymal stem cell. That's kind of a long word. It's just, mesenchyma means connective tissue in an embryo. So these cells that are kind of just mulling around there, they're called mesenchymal stem cells. And they are found in almost every tissue of your body. Video of this young lady here who was born, plug in, is vulnerable, should be updated. Well, and I guess we won't do that.
Dr. B, if you use Google Chrome, it should work.
Huh?
If you use Google Chrome instead of Firefox, the video should work. If you just copy and paste the link.
Google Chrome instead of Firefox, it should work. Okay. I'll keep talking and maybe play with it and see what happens. Okay, long story short. Chloe was born, she was normal, then over 6 months it was very clear that she was not reaching her guideposts of development. She could not walk. They called it the Chloe shuffle. On the video, there's a video of her, she's on her backside, essentially just pulling herself along, shuffling. And parents were unsure what to do. They took her to the doctor. The doctor referred them to a neurologist. The neurologist did some brain scans. It was clear she had cerebral palsy. They were devastated. They weren't sure what to do, but then they heard about an experimental procedure using Chloe's cord blood that was being infused into her brain. They decided to take the chance. They had stored her cord blood. When the cord blood was then infused into her brain, something really dramatic started to happen. Instead of not babbling, she now started to babble and even started to form words. And then she stood up. Then she took her first steps. Now this girl is walking and talking. She is not completely normal, but she's near normal. And the hope is that as she grows and develops, that she will become more normal. But this little girl who would never talk, who would never walk, now is able to both walk and talk through stem cell treatments that came from her own cord blood. I'm just going to give you just a quick, just a little list of some of the things that these types of stem cells have been able to treat. This doesn't mean that these patients have been cured, but essentially all of these patients have had their conditions improved by application of these stem cells. In fact, corneal scarring, you have a stem cell population called the limbus cells population that's on the cornea. And transplants of corneal stem cells can actually take corneas that are clouded and scarred and they'll make them clear so that you can see again.
Dr. Radovich, I've got the cord blood miracle on my Apple. It's a YouTube if you want to see it.
I want to respect the time check. I don't think I'm going to have time, so I will. My students all know I talk too much, so. Just take a look at some of the lists of some of these clinical trials that are going in. These are really life-changing, devastating sorts of problems. Parkinson's disease. When you need new blood vessels, growing new blood vessels in tissue in culture. Anthony Atala's lab at Wake Forest has been able to not just grow new muscle and new blood vessels, but his group has actually been involved in a revolutionary new science called tissue engineering, making new tissues using stem cells, and again, not embryonic stem cells, using somatic umbilical cord and other types of stem cells. Heart treatments. People who've had heart attacks have been treated with stem cells from their own bone marrow, and in some cases stem cells from their own fat, or in some cases, because mesenchymal stem cells tend to not be detected by the immune system, from mesenchymal stem cells from someone else. And this is just a skimming of some of the clinical trials. All of these trials have been very successful. In fact, the Repair AMI study had over 200 subjects. It was a double-blinded, placebo-based study that showed remarkable increases in heart function after implantations of their own bone marrow stem cells. Barry Brown was a personal trainer. Working out was his life. Then one day there was some pain across his back. He didn't know what it was, but he ignored it. Then 2 years after that, he discovered when he was on the treadmill, he was out of breath. He couldn't discover why. So he went to his doctor, and it was very clear that he had had a silent heart attack. He had extensive scarring in his heart. He would never work out again. He was told by his doctor, if you work out, it will kill you. So he enrolled in a study called the Prometheus study. Joshua Hare and his team were using mesenchymal stem cells from bone marrow and treating people's hearts who had just had a heart attack with them. And 3 years after his treatment, Barry Brown ran a half marathon. And you can Google this on YouTube, and you can see him running the marathon. This is remarkable recovery. And the Prometheus study was published several years ago, and Joshua Hare has done several other studies showing that mesenchymal stem cells can not only recover heart tissue, it can shrink the heart scar and increase heart function. Scleroderma. This is a disease essentially where your immune system starts to attack your skin and blood vessels. You get extensive scarring in the skin, and it turns your skin to stone. Then it turns to your internal organs. It usually starts after the skin, the lungs, then the heart, kidneys, the gut. It just turns your body into stone, essentially. Both of these people, the Amy Daniels story, she talks about when she was diagnosed, you can look at her skin, you can tell that her skin has kind of been through the war, but she wrote a letter to her family telling her husband, I'm going to die. It's okay for you to remarry. The girls need a mom. It's okay for you to move on. And she heard of an experimental treatment from Richard Burt using, again, her own bone marrow stem cells. Mesenchymal stem cells have the ability to suppress a rogue immune response. And according to Ms. Daniels, a few hours after she had her infusion, her skin started to soften. Her lung capacity also started to come back. Her disease now is in complete remission. She should have been dead in 6 months. This other chap here, a construction worker, Tony Underhill, if you look at his video, he's got this beautiful southern twang about him. And he said his skin was so bad, you could have tried to stick a knife in it and the knife wouldn't have gone in. That's how hard his skin was. He made a near complete recovery, again, with bone marrow transplant, using stem cells from his own artificial kidneys, using tissue engineering. They're not big enough to work. It's a sizing issue now. That liver, you can actually transplant that into a mouse, and it will work as a liver. And in fact, that mouse now will respond to drugs the way a human would. So it becomes a perfect model for drugs in humans. If you want to replace a human liver, what do? You need to make it bigger. So it's a sizing issue. Maybe someone in this class will go into this field and actually solve some of these problems. All of these were made with stem cells from your body or from umbilical cord cells. So in conclusion, I'd simply like to say that there is a revolution going on in medicine, and it doesn't include embryonic stem cells. Do embryonic stem cells have a role? Yes, they do. In the laboratory with drug testing, drug screening, things like that. We don't need to be murdering embryos. And yes, I'm going to use that word, murdering embryos, because these are young human beings. Guess what, gang? Everybody here was once an embryo. Everyone was once a Dan embryo, a Renee embryo, a Kristen embryo. You were known by God at that time. To essentially take you and dismember you to advance science is murder. And I'm not going to pull any punches about that. That's what it is. So I will now take your questions, and we have books for sale, and they're at a discount even. And there'll be a signing after the seminar. But now I'm sure some of you